Somewhere in the eastern Pacific, roughly 200 meters below the surface, it is completely dark. There is no sunlight here, no bioluminescent lure from a predator to draw attention. Just darkness, pressure, and cold. Inside that darkness, a school of Humboldt squid is hunting. These animals are not small. Adults can reach 2 meters in length. They weigh up to 50 kilograms. They hunt in groups that sometimes number in the thousands. What makes the coordination of this hunt possible in an environment with no light is one of the stranger problems in marine biology. The solution appears to involve flashes of bioluminescent light, not as camouflage, not as lures, as signals. Humboldt squid, Dosidicus gigas, are capable of producing complex patterns of light using chromatophores, specialized pigment cells, across their bodies. Chromatophores expand and contract rapidly, creating visible flashes. The speed of these color changes is faster than most fish can track. Squid can see them clearly. What researchers began to suspect, based on underwater video footage captured by remotely operated vehicles, was that these flash patterns were encoding information. Not random bioluminescence, not simply a side effect of nervous system activation under stress. Intentional, patterned, information-dense signals. The idea was controversial when first proposed. Chromatophore-based communication had been studied in shallow water cephalopods for decades. But applying the communication framework to coordinated group hunting behavior in deep, dark conditions was a significant interpretive step. The evidence for it built slowly through observational analysis of ROV footage. Research teams reviewing hours of deep-water ROV footage noted that Humboldt squid flash patterns were not random during hunting events. Individual animals produced rapid, alternating dark and pale flashes. Neighboring squid responded within milliseconds. When one squid entered an attack posture, a characteristic flash pattern preceded the attack. Animals around it shifted their own body orientation. The school changed shape without any single individual apparently leading the movement. The formation adapted continuously, coordinating approach angles on fish prey. Fish caught between converging squid formation lines had no effective escape path. The predation efficiency appeared significantly higher in coordinated group events than in individual attempts. Individual Humboldt squid attempting solo prey strikes on anchovy or sardine have low success rates. Group formation hunting, where the chromatophore signaling appears to coordinate simultaneous attacks, had much higher documented prey capture rates in the ROV footage. The specific chromatophore flash patterns associated with hunting coordination have been categorized. A dark slash pattern, a rapid dark band traveling across the mantle, appears associated with an attack initiation signal. A pale flash, rapid full-body paling, appears associated with a retreat or hold signal. Intermediate combinations seem to encode different states. This is not simply yet a fully decoded language. The vocabulary is partially mapped, not fully understood. What is functionally established is that the flash patterns during coordinated hunts are non-random, and that conditions disrupting clear bioluminescent signaling reduced coordination effectiveness. Squid removed from a group and isolated still produce flash patterns during simulated attack contexts. Returning them to a group context with active flashing animals restored coordinated behavior faster than in control groups with no flash activity. The signal is functional, not incidental. One of the more unusual features of Humboldt squid hunt coordination is the absence of identifiable leaders. The formation responds to flash signals from multiple individuals simultaneously. When one animal changes position, others adjust relative to it. When that animal adjusts again, the group reshapes. The practical result is a distributed coordination system. No individual carries authority. The formation is an emergent property of many individuals, each responding to the signals of those around them. This kind of distributed coordination has analogs in starling murmurations and fish schooling. But those systems work by visual tracking of neighbor position. In the mesopelagic zone, where Humboldt squid hunt at depth, visual tracking of neighbor position depends entirely on bioluminescent production. The squid can only see each other because they emit light. The chromatophore signals do double duty. They make the animal visible to neighbors, and they carry information about behavioral state. Two communication functions in one system. The predation impact of Humboldt squid on mesopelagic fish populations is ecologically significant. At peak densities, Humboldt squid aggregations consume millions of metric tons of fish per year. In the California current system and the Humboldt current system along the South American coast, squid predation pressure on anchovies, sardines, and lanternfish is a major structuring force in food web dynamics. The coordinated hunting formations multiply that predation efficiency. A formation that can encircle and trap a fish school simultaneously from multiple angles removes most of the prey school's escape options. Individual fish flight responses, which evolved against single predator threats, are much less effective against a coordinated multi-angle attack from a formation of hundreds of large cephalopods. This asymmetry, prey defense evolved for solo predators, deployed against group formation hunters, is part of what makes the squid's predation efficiency so high. It also helps explain why the prey biomass consumed by these animals substantially exceeds what would be expected from their individual hunting performance. The formation is not just coordination, it is a force multiplier. Most of what is known about Humboldt squid hunting behavior comes from ROV footage and bioluminescent sensors deployed at depth. Direct observation of the hunts is limited by the extreme environment. ROV presence may alter squid behavior. The footage represents a partial window into what happens in these deep midwater zones. Researchers have been careful to distinguish between observed behavior and inferred coordination. The chromatophore signal patterns observed during coordination events are documented. The specific cognitive or sensory processing underlying the responses to those signals is inferred from behavioral outcomes, not from direct neural measurement. Cephalopod neuroscience at depth remains one of the more challenging frontiers in marine biology. Whether Humboldt squid hunting coordination constitutes intelligence is a question that requires defining the term. The formation emerges without individual foresight, planning, or communication of intent in any human sense. Individual animals respond to local signal information. The global behavior emerges from those local responses. This is the same mechanism that produces collective intelligence in ant colonies, bee swarms, and other superorganisms. Calling it intelligence at the group level depends on what threshold of adaptive behavior is considered sufficient. The coordinated formation consistently outperforms individual hunting. It adjusts dynamically to prey school behavior. It reorganizes when disrupted. By most functional definitions, that is a system worth calling intelligent. Whether the individuals themselves are intelligent or whether intelligence is an emergent property of the group is a question that the squid's behavior cannot answer by itself. It is a question that the study of collective behavior more broadly has not fully resolved. Bioluminescent signaling is not the only communication channel available to Humboldt squid. Chemical signaling through skin secretions and olfactory reception is possible, but poorly documented in this species. Some researchers have proposed that stridulation, mechanical sound production, may supplement visual signaling in some cephalopods. The relative weight of these potential channels in coordinating Humboldt squid hunts is not established. The visual chromatophore system is the best documented coordination channel. It is the one for which observational evidence from hunting events exists. Other channels remain speculative for this species in this behavioral context. Bioluminescence in deep-sea animals is typically discussed in terms of camouflage or lure. Counterillumination, matching ambient downwelling light to reduce silhouette, is used by many mesopelagic species. Lure bioluminescence in anglerfish is iconic. The Humboldt squid case represents a different functional axis. The same biological system, light production by the skin, serves communication during coordinated predation. This is not a minor variation. Communication and camouflage are functionally opposite goals. Camouflage requires reducing the signal. Communication requires sending it. The fact that the same biological machinery can serve both functions depending on context reflects the general cephalopod pattern of extreme behavioral flexibility applied to a single physical system. Squid are among the most studied animals in neuroscience for reasons that include this kind of multifunctional biological complexity. The chromatophore system is controlled by the optic lobe, the skin is wired directly to the brain, and the signaling repertoire can be adjusted within a fraction of a second. No other animal group has a signaling system that is simultaneously this fast, this complex, and this directly under central nervous control. The hunting formation in the dark water is one expression of that system. One function among many, but it is the function that has the most direct ecological consequence. The fish schools in the mesopelagic zone are not adapted to it. And that asymmetry in the deep dark water is what makes the squid's predation system work. Humboldt squid populations have shifted significantly in range over recent decades. Warming of the California current system and oxygen minimum zone expansion have altered where these animals aggregate. Range expansions into the Gulf of Alaska and other northern Pacific regions have been documented since the early 2000s. These range shifts bring coordinated Humboldt squid hunting formations into contact with new prey communities. Salmon, rockfish, and other commercially important species in expanded squid range areas are now experiencing predation pressure from animals whose coordinated hunting system those prey species have not previously encountered. The ecological consequences of this are actively being studied. It is a case where an evolved coordination mechanism, originally developed in a specific ecosystem context, now operates in new ecosystems without the coevolutionary history that allows prey to adapt. The darkness is the same. The ecological story of this animal's range expansion is still being written, one formation at a time. The flash signals are the same. The prey, however, have never seen this before.